So you can see my title, Coherent Nonlinear Raman Microscopy. And this is interesting because it is a label-free but chemical-specific way to image. And I will explain in this lecture exactly what I mean. So this guy, Anthony van Leeuwenhoek, Dutch, is considered the father of optical microscopy. He was a lens maker who made the best lenses. He was able to get about 200 times magnification. He discovered bacteria. He was the first one to observe red blood cells in spermatozoa. And this really was a complete revolution for people because people had no idea that there was such a microscopic world. And this, of course, changed our conception of what it is to be an animal. It can change our conception of disease and was really a revolutionary step. So what does it mean to image? What do you need if you want to see small things? Well, you need, first of all, spatial resolution. You need to be able to see that something of a certain size. And you need also what's called contrast. And contrast is important. Contrast is why on a football team, we have the different teams wear a different color of jersey. You can imagine how it would be to watch a football game if all the team members had the same color jersey, green, just like the grass on the field. And it would be much harder to see what is going on. It's not because of spatial resolution, not that you cannot see them, but it's rather that you have no contrast. So to see, we need two things. We need spatial resolution and we need contrast. We need visible light. Why? Because infrared light cannot really penetrate biological material because water is like a black hole for infrared radiation, absorbs everywhere. And as you know, UV light also is absorbed by biological material. This is why we wear sunscreen. This is why UV can damage DNA. So in between IR and UV is visible light. So we need to use visible light really for imaging. Cells are small, and we need to be able to see things on the order of a micron or less in order to see inside a cell. And here's the problem I want to discuss with you today. Unfortunately, the intracellular organelles do not come color-coded for our viewing pleasure. They're not colored, and color is what we call contrast in the visible. So that's a problem. People use modern microscopes. This is an example of what's called a confocal laser microscope to do 3D imaging of cells. And usually it's based on some dyes or stains which are used to color things inside a cell. And if you go online, you can find pictures like this. This is from a cow. These are artery cells, and these are beautifully colored, but this is not natural. They are artificially colored. The nuclei are stained blue with a molecule called DAPI. The microtubules are stained green by some antibody, and these protein filaments, actin filaments, are stained red by another molecule called phalloidin. So this allows you to make beautiful pictures. This is what gives you the contrast. But we don't really know how these exogenous, these external dyes and stains will affect the complex biochemistry of living cells. People argue about it. Some people say it's no problem. Other people say it could be a problem. We know that certain chemicals are causing cancer, for example. So if you want to study live cells as they go about their business, living, maybe we shouldn't add anything. But if we don't add anything, how do we get contrast? So fluorophores are the dyes and stains that we add so we can get contrast. There's problems with them. They may interfere with the life of the cell. They're also prone to damage by shining light. They can undergo chemistry we call photobleaching and photodamage. they may stick to the wrong thing, and in which case you're targeting the wrong thing. And because we don't know what they're doing inside living cells, these are certainly not applicable to humans. You cannot stain a human being so you can see what's going on inside their tissues. That's not allowed. So we know cells are made of molecules, and we'd like to see where these molecules are inside of cells and what they're doing. Can we achieve molecule-specific imaging? Well, I'm going to say let's try to avoid these external dyes and stains. Well, fluorescence, which is electronic spectroscopy of organic or biomolecules in solution, is not very specific. It's not diagnostic. Many things can fluoresce if you shine UV light. DNA, proteins, and so on, they can all fluoresce, but we cannot tell them apart from their fluorescence. So it's not specific. There are other kinds of imaging you may have heard of, like MRI, magnetic resonance. This could be specific, but it does not have the spatial resolution because effectively the wavelength is too long. The spatial resolution MRIs may be a millimeter, but we need something better than a micron in order to see inside a cell. So that's not going to work. Well, what this lecture is really about is that molecular vibrations actually are molecular fingerprints. This is the field of spectroscopy, and almost all of analytical chemistry, pharmaceutics, etc., are based on recognizing molecules, identifying molecules from their vibrations. This is very well known and is a standard technique. So can we take advantage of this idea that somehow we can tell molecules apart by their set of vibrations? Maybe that's a way to go forward. Well, vibrational spectroscopy is in the infrared spectral region. It's also called infrared spectroscopy for this reason. And depending on the mass of the atoms, the frequencies of vibration can either be high if they're very light atoms like hydrogen, or very low if they are slow bending vibrations of some molecular backbone. So in fact, this field of spectroscopy can really identify the shapes of molecules. So this maybe is a good idea. Maybe there's a way to use molecular vibrations as a label-free way to get contrast. But there are problems with this idea. The problem is that infrared radiation is strongly absorbed by water, and cells and tissues, biological material is full of water. As you know, probably 90% of your body is water, and water absorbs like a black hole. So we won't be able to see inside a cell using infrared light. And there's another problem, which is the infrared light is long wavelength. The wavelength is typically somewhere between 3 and 10 microns. And the spatial resolution we can achieve in optics is something typically on the order of half of the wavelength of light that you're using for imaging. So if we want to see something that's less than a micron, you can see that this is not going to work. We won't be able to see things that are small enough. So for these two reasons, infrared microscopy is not going to work very well for biological materials. So nice idea, too bad it doesn't work. Well, I want to tell you that there's another type of spectroscopy which is also sensitive to molecular vibrations. It's called the Raman effect, and it's based on the scattering of light rather than the absorption of light. And what this means is you can use visible light rather than infrared and still get information about vibrations. And the idea is that you shine light in of this blue color, and what you see is scattered light is shifted in color, and it's shifted by amount that is equal to a molecular vibration. Raman won the Nobel Prize for the discovery of what we now call the Raman effect. So it also is a spectroscopy that probes molecular vibrations. How does it work? Well, here's an idea. I have laser light, which is oscillating at a certain frequency omega, and I have a molecule, here's a molecule, and it's vibrating. And it has a vibrational frequency, omega k, which is different from the frequency of light. Because it's charged, it's like an oscillating charge. So what this means is that, in fact, the interaction of light and matter means that I will have something that has a product of two frequencies. One is the vibrational frequency, and the other is the frequency of light. And when I multiply two frequencies together, I get, as you know from trigonometry, I'll get sum and difference frequencies. The difference frequency, which is shifting to a smaller frequency, a redshift, is called Stokes scattering. And the sum frequency, which is uh, shifted to the blue, higher frequency, is called anti-Stokes for historical reasons. But basically, what you can think of is that the molecule essentially is like a modulator. It's adding sidebands to the frequency of incident light. And the shift of the sidebands is a direct measure of the modulation frequency. In this case, the modulator is a molecule. So in this picture, if I take, here's a solid, sulfur, and here's my laser light, which I have greatly suppressed with a filter. And if I look very carefully with a spectrometer, I will see that there are side bands of this laser light that are shifted by these amounts here. And those are measured in what are called wave numbers, which is related to the vibrational frequency. So directly from this scattering of light, this inelastic scattering of light, I get information about the shifts which are directly related to vibrational frequencies. So this is nice because I can use visible light, which is what we would like to use in biological imaging, in order to get information about vibrations, which allow me to identify molecules in a very natural way by their vibrational frequencies without adding any dyes or stains. So if I was to take a cell and grind it up and put it into a Raman spectrometer, I would see these kind of vibrations in a Raman spectrum. So I'd see CH3, which is due to proteins. The CH2 is due to lipid membrane. I'd see things related to DNA, etc. So this looks quite promising. If I could tune my Raman light to image, let's say here, phosphate, which is DNA, when I make an image, then I would see the spatial distribution of DNA or proteins or lipids. So this looks very promising. Can we use the chemical specific information of the Raman spectrum as the contrast agent in microscopy? And you can. This is called Raman microscopy. You can buy a Raman microscope if you had a lot of money. And here's an example. This is a plant cell. And you can see if you look at different regions and you look at the Raman spectrum, you can see that they're different. And this is what's called Raman mapping. And it works. But there's a problem. And the problem is that the Raman effect is very, very insensitive. The molecule is a very small modulator. It doesn't give much signal. So it's very, very insensitive. If there's any fluorescence from the sample, which there could be, then the fluorescence will be much, much larger than this Raman effect. And it will be completely overwhelming your signal. You won't see this tiny amount of light from the Raman effect. And even if there's no fluorescence, because the signals are so small, it can take a very long time to generate a Raman image. You just need enough statistics. So this means it's very slow. It can take sometimes hours to get a Raman image. But there's a problem if you want to study live cells, because live cells are not static, they're dynamic, they move. A cell will divide on a time scale, let's say 20 minutes. So you need to be able to image faster than this if you want to make kind of a movie of live cells going about their business. So this idea of Raman microscopy is a nice idea. It's label-free, chemical specific, but it's not going to work because it's too slow. It's too insensitive. So nice idea, but it doesn't work. What are we going to do? How can we possibly proceed? Well, this is the main subject for today, and it's a subject called nonlinear optics, and has to do with the idea that if I shine light on a material, I have normally what's this linear response. E is the electric field of light. So this first term here is what we call the linear response. And if you analyze this linear response, it contains things that you know very well. These things are the refractive index and the absorption coefficient. And this is almost everything we know about spectroscopy is based on this linear response. However, if I increase the E the electric field, it turns out that there could be a nonlinear response, something that goes like the square and the cube. These higher order terms were not even considered until the laser was invented, because before the laser, people never imagined that the electric field of light would be strong enough to have any nonlinear response. So this only came with the invention of the laser around 1960. So as I mentioned, this first term, this linear response, is known very well and gives us the things we know. Almost everything we know about spectroscopy is contained in that linear response. And when the E is very small, E squared and E cubed are even smaller, and so these nonlinear terms are negligible, and people did not consider them. However, if I had a way, like with a laser, to turn up the electric field, then these nonlinear terms become apparent. In fact, they can become dominant. You could see at some E, E squared will be bigger than E, right? These will grow in a nonlinear fashion. So this is actually interesting because it gives many new opportunities. Different kinds of spectroscopy become possible when you have these nonlinear responses, which are not possible when you have a linear response. And this is partly what my lecture is about today, is talking about the new opportunities that come from this nonlinear response of matter to light. And you can understand it like a harmonic oscillator. So when I apply oscillating electric field to electrons in matter, I'm basically shaking them. The electrons are shaking up and down. Like a spring, there's a restoring force U, like a spring constant. And the question is, what is the nature of this restoring force? If the restoring force is linear, like for a spring, F equals kx, then I get all of linear optics. But you could imagine that if the spring constant changed with extension, I might have to have a nonlinear response. That's called anharmonicity. For example, something goes like the square of the cube. Then these would be nonlinear responses in the shaking of this oscillator. And this turns out to give new frequencies. So the polarization of the material includes the fact that the electrons are being shaken. And the electric field inside a material, as you may know from Maxwell's equations, contains not only the electric field you apply, but also this polarization response of the matter. And if the matter is responding in a nonlinear way, then I have to include this in my description of light propagating in a material. So there's a few equations, but the main point is that if I include these higher order intensities, this nonlinear polarization, then I can describe them as interactions between these electric fields that go like the product of two or the product of three. The linear term is already very well known. So what are these nonlinear terms and what can they do for us? What do they mean? Well, some of these things you probably have heard about. There are second order nonlinear optical effects that go like the square of the electric field. Probably the most famous one is second harmonic generation. But second harmonic generation is a special case when the two colors are the same. The more general case is when the colors are different and you get sum and difference frequency generation. And there's some other effects known as the Pockels effect. There's also third order effects where the response goes like the cube of the electric field. And these are also known as third harmonic generation. There's something called Fourier mixing, something called the optical Kerr effect. And there's a particular one, which I'm going to talk about today, which has this acronym CARS. And CARS stands for Coherent Anti-Stokes Raman Scattering. Um, in nonlinear optics, there are processes that are called parametric. And basically, that is when you start and end on the same state. And this is described by a real susceptibility. And there are non-parametric processes where you end up in a different state, and these are described by a complex susceptibility. You could basically think of these parametric processes as a nonlinear version of the refractive index, and these other ones are like a nonlinear version of the absorption coefficient. You can think of them like that. Here's an example of second harmonic generation, which you probably heard of. I send in this color omega 1, which is red, and somehow through a medium, a crystal, it turns out, that has certain symmetry, the two kind of get multiplied together, and I get something at twice the frequency, blue light. In fact, that's how nonlinear optics was discovered. It was discovered by accident. Somebody put a quartz crystal in front of a laser beam, and they saw blue light out from red light in, and they said, how did that happen? And discovered second harmonic generation. So there are different types of these second order nonlinearities. I mentioned to you second harmonic generation, but there's also sum and difference frequency generation, etc. But the main point is that you have two electric fields, if you like, two colors of light, E1 and E2, and somehow they get mixed together by this nonlinearity. What about third order? Well, third order means that there are three electric fields, E1, E2, E3, which have different colors. And there are many different ways you can multiply these together, as you might imagine, right? You could have 3 times this frequency or one of those, that'd be third harmonic generation. But I can also take different products. One of them, which we're going to talk about today, is where I have the second harmonic of one where I take the difference with another one. So 2 omega I minus omega j. And this is the term that is relevant for this CARS, this coherent anti-Stokes Raman scattering technique. It's a third order nonlinear response that depends on frequency differences between two colors. There's one other interesting point about nonlinear optics, which is relevant for biological imaging. So there's something called the diffraction limit, which basically says that it's hard to see something that is, let's say, smaller than half of the wavelength of light. And it has to do with diffraction. The fact that if you try to make light smaller and smaller, it starts to diffract. So if I put light through a very small aperture, I get a diffraction pattern that looks like this. And you might expect that your spatial resolution might be measured by the size of this smallest disk here. That's called the Airy disk. And it turns out that that diameter d is something like the wavelength divided by 2 times what's called the numerical aperture, which has to do with the focusing. Numerical aperture of about 1 is about as good as you can get. So you can see that the best spatial resolution that you will achieve is something like half of the wavelength of light. But in non-optics, we have something different going on. The signal is generated only by the very peak of this little Gaussian here. So in other words, the signal is not generated everywhere, but only at the very peak. What this means is that the signal, the nonlinear signal, comes from a much smaller area than does the overall focus. And you can see that this area is smaller, which means that the spatial resolution will be better than this diffraction limit. So nonlinear optics also offers an increased spatial resolution compared to linear optics. Okay, let me talk about the main subject for today, which is this CARS technique. It's a third order parametric process. You can think of it as a nonlinear optical version of Raman spectroscopy. Because it's nonlinear, we would like the electric field to be high, which means it's really best used with short pulses. Why? Because in a short pulse, the pulse is very, very skinny and very tall. The height is the intensity of the electric field E. And so the taller it is, the more nonlinear response you'll get. So what do I require? Well, I require 2 input colors. So I show them here as green and yellow. So I told you that it goes up and then down and then up again. And the signal that I generate is actually this blue shifted, this blue arrow here, which is called the anti-Stokes radiation. So what do we measure is actually this blue light as a function of the difference between these two, yellow and green. And you can see that when the difference frequency matches some vibrational level, I'll have a resonance. And that resonance is an indication of the molecular vibration. So in other words, this is a stimulated version of the Raman effect rather than a spontaneous version, which I showed you before. And this is the big difference. Because it's stimulated, it is at least 10,000 times, some people might say 100,000 times more sensitive than the spontaneous Raman effect. So this is a game changer. This factor of 10 to the 4, 10 to the 5 means that we can actually now do imaging using the Raman effect much faster, means we can make real-time movies. What are the advantages? It has enhanced 3D spatial resolution because of the nonlinear effect. It has selectivity because I choose the difference frequency between these yellow and green colors. I choose the difference frequency to match some molecular vibration, but I get to choose. So it means I can tune on to a given Raman mode. And that gives me now the chemical contrast. It's label free because I don't need to add anything. I don't need to add any dyes or stains. There's no linear absorption. I can choose those yellow and green colors so that they're not directly absorbed. It's only the difference frequency that counts. So it means that I'm not going to burn the sample. I'm not going to heat it. It's insensitive to fluorescence because I can choose colors that are not absorbing, and so it doesn't fluoresce. And also, the signal, as I said, is 10,000 or 100,000 times stronger. So it actually means it can even be stronger than fluorescence. And important for biological materials is that I can go to the near infrared. I can choose these colors so that they're in the near infrared, let's say 800 to 1,000 nanometers, so that I can penetrate more deeply into biological material. If you hold your hand up to the light, it looks red. The reason why is because red light passes through biological material more easily than visible light. So these are the reasons why it's of interest. Now, there is a problem that I have to tell you about. And the problem is that this diagram here, this third order response, this up and down, which has a difference frequency which matches some Raman vibration, and then it has one more up, and then what I measure down is this blue arrow. That's my Carr's effect. This is what I would like to have happen. And when I have a resonance, I will see my molecule of interest. The problem is, at least in Canada, is that molecules are stupid and they don't know that you want to do this. And they can also do this. And all I do here is I change the time ordering of these two arrows. Instead of up, down, I go up, down, down. And this blue arrow and that blue arrow are the same length. So therefore, they're the same color. This has no resonance. You see, this one is not sensitive to this vibrational level here. So this is a non-resonant term. You might say, well, resonance is a good thing. Probably the resonant term will be bigger. That could be true, but it may be that the molecules, the resonant molecules which you study are there in much lower concentration, and everything else inside the cell, all the water, for example, will be non-resonant. So this term can actually be much bigger than that one in real life. And that's a problem because I cannot prevent this from happening. I cannot tell the molecule which order they should absorb the photons. These will both happen. And this will reduce our contrast because this will give signal everywhere and this will give signal only for the molecules of interest. So that's a problem. How can I differentiate these? It also means that this interference between these resonant and non-resonant terms gives you funny line shapes. So here's a molecule of nitrobenzene. This is the Carr spectrum. What I can show you is that the peaks are in the wrong place. They're shifted from where they should be according to the true Raman spectrum. And this has to do with this interference effect. So that's a pain. This Carr's microscopy idea sounds nice, but there are distortions that are associated with it. So this non-resonant background is a problem. We would like to get rid of it. It reduces the contrast and it's not molecule specific. So what should we do? Well, the first thing you might want to think about is you want to make sure that all your photons are actually on resonance. So let me give you an example. Here's a Raman peak that was measured by somebody. You could look this up in a book, and it has a certain line width. I'll use wave numbers as a line width. This would be a carbon-carbon stretching frequency. This could be in some polymer or some plastic material, something like this, that has carbon in it. So you would have these frequencies here. So the best you would do, you think, is, well, I'm going to choose a laser bandwidth that has the same bandwidth as my peak, so that if I tune onto that resonance, all my photons are resonant. And this would be the best I could do. And if you were to come with a shorter pulse, like a femtosecond laser pulse, a femtosecond pulse has a much broader bandwidth. You say that's no good because most of the photons will be off resonance. They won't be on that peak. And so this would be a bad idea. The problem is that we would like to use short pulses because the nonlinear response will be higher. So this sounds like a fundamental dilemma. We should use longer picosecond pulses so that they match this line width. So we are on resonance, but we would like to have the intensity, the electric field of the femtosecond pulses. But it seems that it's going to be off resonance. What can we do? Well, in other words, how can we get vibrational resolution when we have laser pulses that are too broadband? How can we deal with this? And what I'd like to emphasize today is a fundamental difference between linear and nonlinear optics. And it has to do with something called the optical phase. So I first need to tell you what is a short laser pulse. So a short laser pulse we can only make by synthesizing together many different colors. So the way I describe a femtosecond laser pulse is I have many frequencies, if you like, many cosines, and I tell you the frequency, the amplitude, and the phase of the different colors in the pulse. And if I synthesize them together by here a Fourier transform, you can see I can construct a pulse in time. But the shape of that pulse depends on the amplitude and the phases. So that's how I understand what a short pulse is. I understand it in terms of the amplitudes and the phases of all of the individual colors that make up that pulse. How do I describe the phase? Well, what people typically do is they say, well, there is some phase and it's going to vary slightly across my pulse. So I'm going to do what mathematicians do, which is I'm going to do a Taylor series expansion of this phase, this 5 omega. This term here in the exponent, I'm going to do an expansion in the Taylor series. There's a linear term, which is basically the same thing as a time delay, but there could be a quadratic term, this quadratic term in the phase, and this is something that's called a linear chirp, and this is what I want to talk about. What is a linear chirp? Well, you're kind of familiar with it. It's like the Doppler effect that you hear when a fire truck goes past your house. It's a frequency that increases or decreases linearly with time. So you can see here that the frequency, which is measured by the distance between these maxima, is larger here and it gets smaller. This is what we would call a chirp pulse. The frequency is changing linearly with time. And I can describe that mathematically with this chirp parameter here. So why is that interesting? Well, I mentioned to you that the problem with femtosecond laser pulses is that the spectrum is too broad and we're not going to have good resolution. We won't have the contrast that we would like. Contrast means that we tune onto a vibrational peak and identify the molecules by their Raman spectrum. But if I don't have a spectral resolution, I cannot tell these different modes apart. So let me do something simple here. I'm going to chirp one of the laser pulses. Let's say, remember there's two laser pulses, a green and a yellow. I'm going to take one of them. I'm going to stretch it out in time. Here I've stretched it out. So I've chirped them. So you can see that the frequency is changing linearly as a function of time. The red colors are here and the blue colors are there. But I'm going to keep the other laser pulse. I'm going to keep it short. What I can tell you is that the Raman effect is an instantaneous effect. It's a scattering effect. It's not the absorption and then emission of light. It's an instantaneous response. So if my other laser pulse, let's say the green one, is at this time here, it will only see the colors that are there at the same time. So it'll only see these yellow colors. It will not see the red ones or the blue ones because they're not there at the same time. So that means, in fact, that I've gated out, if you like. a smaller part of this whole spectrum. So that sounds promising. And in fact, I can even do something nice, which is I can actually tune across the Raman spectrum just by changing the time delay. By changing the time delay, I can overlap instead with the yellow, I can overlap with the red part. So this sounds kind of nice. So what happens if I do that? Well, let me show you. This is on my microscope, and I'm playing with this idea of chirping the pulse. So if I don't do any chirping, and I put a drop of methanol, which is a liquid, onto my microscope, and I scan across the spectrum, I get these open circles. You can see that there's some kind of resonance there. The signal goes up and down, but I would not be able to tell you that this is methanol because I have no spectral information. But if I chirp the Stokes pulse the way I described, you can see I can now start to see that indeed I can identify some peaks there. So now I can say, oh, looks like methanol. So chirping of this laser pulse actually gets me spectral resolution that is better than the bandwidth of my laser pulse. Can we do better? Well, if chirping works once, why don't we do it twice? What if I chirp both laser pulses? But I'm going to do so in a very special way. Let's take the center frequency here of, let's say, remember the two colors, green and yellow? Let's take one of them. I'm going to say the center frequency is omega 1, and this one is center frequency omega 2. I'm going to chirp them both. I'm going to stretch them both out as a function of time. But I'm going to arrange them so that the difference frequency, omega 1 minus omega 2, is matching some resonance in my system, some Raman resonance. So these two colors, omega 1 and omega 2, will be on resonance. What about all the other colors? Well, you remember I said I'm going to chirp them at the same rate, which means that I'm going to change the frequency of both colors, but change them at the same rate. So in other words, if I'm going to shift this amount by some delta, I'm going to shift the other laser by the same amount delta. And what that means is that their difference frequency remains the same. So these two colors here also are resonant with the same Raman effect. And the same on the other side here, I'm increasing the frequency by some amount delta. But if I increase the other laser by the same amount delta, then their difference frequency also remains constant. So in other words, even though these colors are very broadband, many colors are here, their instantaneous difference frequency remains constant throughout the entire pulse. So this is what happens when I chirp both of them. Remember what I showed you before? This is when I chirped only one of them, the black lines. When I chirped them both in this way I described, I now have these open squares. You can see that I've really improved the spectral resolution. I can now completely resolve these vibrational modes. And in fact, to prove the point, I'm going to put another molecule here called DMSO. In the black line, you can see that it has a different Raman spectrum. I've not changed the bandwidth of my laser pulses. I've only changed the chirps. And this is the important point. These are still femtosecond laser pulses. What I've done is I've played with their stretching in time. So this is an important idea, and this is called optical pulse shaping. So here's the general principle. Here's the laser pulse, and because it's a laser pulse, it has many colors. So I'm showing you all the colors. And here I'm showing you that all the colors are there at the same time. This idea of chirping is that actually I will keep all the colors, but I'm not going to have them there at the same time. I'm going to make an analogy with playing the piano. A piano has 88 keys. I can play all the keys at once, and this will make a short pulse. But as we know, music would not be very interesting if that's all we did. I can play a score. I can play different frequencies at different times. I can play the keys at different times. So I can do the same thing with optics. I can have a musical score for light. I can play these different colors at different times. All the colors are still there. The 88 keys of the piano are still there. I'm still playing all the keys, but I'm not playing them at the same time. So this idea of keeping a constant difference frequency is I'm going to play 2 keys on the piano. I'm going to put my fingers a certain distance apart. I'm going to go all the way along the keyboard, playing all the keys, but only 2 fingers at a time. I'm going to keep the distance between my fingers the same. So in this way, I'll use all the colors, all of the keys to the piano, but I'll keep a constant difference frequency all the way across. And this is what we are doing, this idea of quantum control or optical pulse shaping. I can describe this in a slightly more mathematical way with this what's called a frequency time plot. So the frequency time plot shows me the time ordering of the frequencies. So here's my pump and my Stokes. I'm showing that they're a little bit stretched. If I just subtract these two, I will have the difference between them, which is this Raman mode. If I stretch them out as a function of time, I have not changed the colors. The frequencies are all there, but if I stretch them out, now this difference frequency becomes very narrow. So you can see that I can stretch this to different degrees, and I can basically choose my Raman spectral resolution as I like. There's one more important point, that to tune across the Raman spectrum now becomes really easy, because the difference frequency is the vertical distance between these two. So here, when the pump is at this time delay, this would be my 2 colors, yellow and green. This vertical distance, if I subtract them, I get this number here, and that is probing 1 Raman level. But if I just change the time delay, you can see that it gets closer together. This difference frequency gets smaller. So it means that the difference frequency has been changed. This is how I actually can tune across the Raman spectrum. This is very nice because it means I don't have to tune my laser. I can record a spectrum just by changing a time delay, which is much easier to do than trying to tune the laser. This, in fact, is the fastest scanning that's available to carbon microscopy, this technique, which is called spectral focusing. To show you how this works, what I'm going to do is I'm going to put a drop of methanol, and what I'm going to do is I'm going to change the amount of chirp, the amount of stretching. So same lasers, same molecules, same microscope, everything's the same. The only thing I'm doing is changing the amount of chirping. And what you can see is as I increase the amount of chirping, my resolution and my signal to noise just gets better and better. So this chirping, this spectral focusing, or if you like pulse shaping, only exists in nonlinear optics. In linear optics, this phase is of no consequence. In linear optics, the only thing that matters is the spectrum. So nonlinear optics gives us new tools, new opportunities for controlling the light. In other words, what we're doing here is we're engineering the light, which is a new approach to microscopy. Okay, so what do we have here? We have a femtosecond laser. We've got a bunch of optics to make the 2 colors we like, and we're going to focus them into a sample in a microscope. We are talking about this 3rd order nonlinear response called CARS. But if I have a 3rd order response, then for sure I also have a 2nd order response, because that would be even easier. Something that goes like the cube would be a small signal. Something goes like the square would be a bigger signal. So it means that unavoidably I'll also have the 2nd order response. So I can actually measure that at the same time, because these will be different colors. So for example, if I shine my light in, I could have 2nd harmonic generation. If my input laser is 800 nanometers, the 2nd harmonic would be 400 nanometers. So in fact, this color is different from the CARS signal, which would be this blue color is 650 nanometers. In fact, I could also have fluorescence from the sample, which would be yet another color. So I can have 3 different channels that I can measure simultaneously. So this is what we call multimodal nonlinear microscopy. I can measure all these things simultaneously by having 3 different detectors. And because they're measured at the same time, I know that the images generated by these different signals will all be registered. They'll all be synchronized. So this is very nice, because what we have is some passive optics in a box. And this, in fact, led to our lab developing the first commercially available CARS microscope with a Japanese company called Olympus. And if you had $1 million, you could buy one too. Okay, it will not surprise you probably that most of the applications of Karst microscopy are in biophotonics and medical imaging. So I want to show you a few examples. But I also want to show you that there are other applications because imaging science is very broadly based. Imaging is also used in material science, in geology, in earth sciences. Imaging is useful almost everywhere. So here's an example, one of the first examples we showed. So this is label-free multimodal cars imaging. And what this is from some collaborators who were doing Raman imaging of a rabbit aorta. They were studying a disease of the heart called atherosclerosis, which basically is fatty tissues are building up inside the heart tissue and restricts the blood flow, and this can lead to problems for the heart. They have been doing what we call Raman mapping, and they want to know whether our technique could help them with their imaging. So what is this? This is a slice of a rabbit aorta, and we tuned our lasers so that we could image lipid droplets, and we colored them in red. So you can see that there are some fat droplets inside this. The blue is second harmonic generation from a material called collagen. Collagen is one of the most important proteins inside the human body, and it's a connective tissue. The green is a material called elastin, and this is what makes blood vessels stretchy. It's called elastin for a reason because it's elastic. This is what allows blood vessels to pump blood, is that they're flexible. And it turns out this gives two photon fluorescence. So what I've done here on the computer is I've taken the three signals from my three different uh, detectors and I've false colored them, red, blue, and green. If you were to look with white light on a microscope, you wouldn't see any contrast at all because these are not colored themselves. I've artificially colored them on the computer using the different signals. And what I can tell you is that our colleagues who are trying to study this problem using um, their Raman microscope would take them 12 hours to get an image. And what I can tell you is with our Karst microscope, we could do the same thing in half a second. So from 12 hours to half a second, that's a game changer. So now we can, as you can see, we can make movies of biological materials, biological tissue. And this is one of the main interests. There's other things we can do. This is a collaboration I had with somebody who is studying the hepatitis virus. And they want to understand how hepatitis infects a cell. Hepatitis is a disease of the liver, and liver cells are called hepatocytes. And so what we're doing here is we're making a movie of live hepatocytes as they go about their business. They are involved with processing fats. That's what the liver does, processes lipids. That's where fats are digested in your body. And these little fat droplets are being moved around by these liver cells. And the goal of this project was to see how the virus actually infects the liver cell. How does it come in and how does it actually work? Could we make a movie of a virus infecting a cell? That was the goal of this project. So that's very nice, and there's lots of applications. But I want to actually show you that there's more applications to this type of microscopy than just in biological imaging, which is sort of the most obvious one. And I'm going to show you some illustrations to biomaterials and also to geosciences, earth sciences, geology. So the first is biopolymers. These are very well known: starch, cellulose, glycogen, etc. These are kind of based on biopolymers are based on glucose, which is sugar. And a polymer means there's many chains linked together. So these polymers based on glucose are known as starches, or more generally carbohydrates. Cellulose is actually the most abundant organic material on our planet. Cellulose is everywhere. It's what makes up plants, and there's lots of plants. The difference is the way in which these things are connected. So there's a lot of interest in biopolymers. plastics, materials based on biopolymers. So can we image these things? Can we learn something about them? Well, I'll show you an example. This is an example of a starch grain. A starch grain is small, on the order of 10 microns. And what it's made up is alternating layers of lipid and uh, carbohydrate, starch. And these alternating layers go from crystalline to non-crystalline. So this is from our lab. This is a multimodal Cars microscopy image of a starch grain. And what you see are these stripes. And the green is second harmonic generation. And what I didn't tell you is that second harmonic generation can only really come from ordered crystalline structures. There's a symmetry requirement that requires something crystalline. The Cars technique, which is tuned here to the lipids, will only see the lipids. And what you can see is that there are stripes, there are regions where there's green and where there's red. And this shows you that the chemical composition of this is varying. We actually did something kind of fun. We actually cooked some potatoes. We cooked these starch grains. And what we did is we made movies of how they behave as you heat them up. This is a very important process in not only cooking, but in the processing of biopolymers in general, because when you cook, when you heat these things in water, they swell, they expand. And so we can make movies of these starch grains showing how they expand. This is as a function of time when they're heating up. This is the Raman spectrum, the Cars spectrum of the potato starch grain compared to a molecule DMSO. So we can make movies of these things, and as I told you, it's a multimodal movie. So in fact, we can do a Cars image and a second harmonic image at the same time. And what you can see, in fact, is that the second harmonic image decays more quickly. What does that tell you? It tells you that the crystalline structure gets lost first before the swelling happens too much. So we learned something by this is something you could never do by any other technique. There's no other technique would allow you to see this in to make a movie in real time like this, because we get simultaneously the second harmonic and the Cars. So this is a nice illustration of something that you can do uniquely with nonlinear optical microscopy, which is the processing of these carbohydrates. Another example is in biofibers. There's a lot of interest in fibers and stress-strain relations in fibers. So here's a single cotton fiber. There's 20 micron in diameter, so it's very small. And cotton fiber is also made of these biopolymers, these carbohydrates. And what I can do is I can tune here in region 1. I can see that there's the Raman spectrum of the cotton fiber. And if I tune over here, I can see the background, which is water. There's a cotton fiber in water. Why is it interesting? Well, because it turns out that if I put a stress, if I put a kink in this fiber, I can see that the Raman spectrum has changed. You can see if I look in region 1 and region 2, in region 2 where I put a stress, I put a corner, you can see that the red and the black um, Raman spectra are different. The ratio of these peaks is different. So this is interesting because now I can get stress-strain relations inside a single microscopic fiber by looking at the Raman spectrum. So that's quite interesting for people that are interested in the mechanical properties of biopolymers. Are there other applications? What about in earth sciences, in geology? Well, here's an example that was presented to us by a colleague, something which is called a fluid inclusion. You probably heard of this because of the famous technique of fracking, which is now a technique where most of the world's natural gas is coming from. And what this has to do with is the fact that there are little gas bubbles trapped inside rock. These little gas bubbles are often hydrocarbons, such as methane. And the interest in the fracking technique is trying to crack these rocks to get out these gas bubbles. So here's an example from Wikipedia showing a little what's called a gas bubble. They call it in geology a fluid inclusion, which show that there's some vapor bubble trapped inside this rock. And people are interested in the conditions and the formation of these gas bubbles in rock. How does this happen? So what I can tell you is that we did the first application of multimodal cars microscopy to the earth sciences. In fact, we have 2 patents on this. So using this cars technique, we could demonstrate 4, if you like, firsts in geology. The first is because it's a high sensitivity technique, we can go very quickly. So we can do what you might call high throughput screening. We can look at lots of samples very quickly, and we're not going to be bothered by fluorescence. So we can look carefully at many, many types of fluid inclusions and do statistics. However, because as I showed you, it's multimodal, we can also get fluorescence and cars at the same time. And this is interesting because higher organics, like heavier petroleum oils, for example, these do fluoresce, whereas methane, a very small molecule, does not fluoresce. So we can see whether there are heavier hydrocarbons present in the rock. We can do spectroscopy because we get Raman information. I can also do spectroscopy inside these gas bubbles and tell you what's inside without having to crack it open. It's a non-contact chemical specific imaging. And finally, because I can do cars and second harmonic generation, I can make correlations between the distribution of these fluid inclusions and the crystalline history of the rock. Geologists think of the Earth as undergoing dynamic processes. There's lots of pressures under the Earth, which lead to the formation of crystals and grain boundaries, etc. How these things correlate to the formation of these hydrocarbon gas bubbles is of interest to many geologists. So here's an example. This is a white field, if you like, just bright light, visible image of a piece of rock. This is quartz, 250 to 500 million years old. And I'm going to look at this section here. I can see that there's some gas bubbles from the optical microscopy, but I cannot tell you what it is. I don't know what it is. So if I do in red here the CARS imaging, I can see, in fact, that there's methane because I can assign the Raman spectrum methane, and I know that these gas bubbles contain methane. There's also some higher hydrocarbons that give fluorescence, and there's also a little bit of second harmonic generation, which tells me there's some crystallinity. So by doing these things simultaneously, I can see the spatial distribution of crystalline structures of the methane and higher hydrocarbons in a completely non-contact, non-demolition way. I can see that the distribution of the methane and the higher organics like petroleum products are not exactly in the spatial location. They're differently located. So I can actually make a map and see where they are in space inside this rock. I can actually say a little bit more about the nature of the heavier oils by the Raman spectrum. I mentioned to you that the Raman spectrum identifies molecules by their vibration. Well, in fact, I can see that the Raman spectrum methane in black and the uh, the the red, which is this region here, are different. They have different Raman spectrum. So this is definitely a higher oil. This is octadecane, which is a test sample to show that in fact this red part here must be a much higher hydrocarbon, much larger hydrocarbon than methane because of the Raman spectrum. And finally, this is something that you could not do in any other way, is because we can get the second harmonic response at the same time as the CARS image, I can actually see a grain boundary. This is a crystalline boundary between 2 zones inside this rock, and where they meet, there's a broken symmetry. And the second harmonic generation is very sensitive to these grain boundaries, these symmetries. And what this shows you is, in fact, these little gas bubbles of methane are collected at the zone boundaries, at the grain boundaries between these, where 2 crystal structures of rock meet is where the gas bubbles seem to be. So in other words, this is new information about the distribution of these methane gas bubbles inside rock that was not known before. So there's an example of an application. This, we argue, is a new field that we call geophotonics. The idea being that if the application of optics and nonlinear optics to biological systems is called biophotonics, then the application to geology and earth sciences should be called geophotonics. So there are many, I chose this example to show you that there's many applications of coherent nonlinear optical Raman microscopy beyond just biological imaging. And with that, I will stop here, um, ending my first lecture. The next lecture tomorrow will be further discussions about nonlinear optical Raman microscopy and another technique which turns out to be even more sensitive, which is called stimulated Raman scattering. But I will stop here and I'll be happy to answer any questions if you have. Good afternoon, everybody. It's morning for me. So I continue my lecture, and today we're going to introduce a difficulty with the technique I described yesterday and its solution. Yesterday, I made an argument to you that that vibrational spectroscopy, particularly Raman spectroscopy, allows you to identify molecules by their shape, by their vibrational modes, and this allows for a label-free natural chemical contrast to be used in imaging. I then argued that coherent nonlinear optics gives you a very great enhancement in sensitivity compared to spontaneous Raman, which is an insensitive technique. And in addition, it offers other types of optical nonlinearities, such as second harmonic generation and two-photon fluorescence, etc. So it becomes a multimodal imaging technique. And I showed you some illustrations yesterday of how this works in practice. So I remind you that this coherent anti-Stokes Raman scattering, this CARS technique, is a Fourier mixing process. It's a third order nonlinearity. Involves putting in 3 photons, a yellow, up, a green, yellow, and a green. And what's measured is this output laser pulse called the anti-Stokes, which is blue. And so this is measured as a function of the frequency difference between the yellow and green, and in microscopy as a function of spatial position. And that's how an image is generated. This coherent nonlinear process is a parametric process. And this means that the signal is quadratic in concentration of the molecule which is resonant. That's good when the concentration is high, but it's terrible when the concentration is low because you lose also like the square. So I argued yesterday the advantages of this technique. It's label free, it's sensitive, it has higher spatial resolution and molecular selectivity, immune to fluorescence, etc. And I gave you some applications in biophotonics and geophotonics from yesterday. I'd like to remind you that I did mention there's a problem. And the problem is that this third order response, this chi-3 response, this third order nonlinearity, is a diagram that looks like this. We want to have an up and a down such that the difference frequency matches some vibrational level spacing, and this is the resonance. Then we have one more up, and this down is the photon which is emitted, this blue color, and that is the signal that we measure. However, this can also happen. I just changed the time ordering of these two in the middle, and then instead of up, down, it's up, down, down, and this color and this color are the same. Therefore, they will interfere with each other because they are completely coherent. They are generated by the same laser pulses, and therefore everything is totally coherent. They will interfere with each other. This interference is a problem. One of the problems is that it gives funny line shapes. You have an interference in a resonant and a non-resonant term, and you get a line shape that looks a little bit like the first derivative of the line. So the true positions are here, and you can see that the fast falling slope is right at the edge there. So you get a funny line shape, which you have to consider, and that's a difficulty. However, there's another problem. And this problem is that there are also spatial distortions of the image, not just spectral distortions. And so here's an example. This is a polystyrene bead in some gel on my microscope. The polystyrene bead, which is plastic, is 1 millimeter in size, 1 micron in size, and it also is fluorescent. And what we're going to do is we're going to tune our CARS lasers, our Raman lasers, to the CH stretch, which this polystyrene has got lots of CHs, so it should give a big signal. But because it's fluorescent, we can do two things at the same time. We can do two photon fluorescence and CARS imaging simultaneously. And something funny happens. What you see is that the beads, the 1 micron beads, are not in the same place when you image them with CARS and two photon fluorescence. There's only one bead, so how can it cannot be in two places at the same time. But yet these two techniques show the bead in a slightly different location. And in addition, maybe you can see in these images here that there's a shadow in front of the bead for the CARS image only, but not for the two photon fluorescent image. So this is a line out through a polystyrene bead showing the signal. And what I've done is I've centered the signal for the two photon fluorescence at 0. And the CARS signal, which is measured simultaneously, right? This is multimodal imaging, so I can measure fluorescence and CARS at the same time. What you see is that the maximum of the CARS signal is displaced, it's behind, and it goes negative in front. There's a shadow in front. So this seems very strange. How can it be? Well, I will remind you a little bit about Gaussian laser beams, which you may or may not have seen before, but it's basically showing that if I focus a laser beam, I do not come to a mathematical point. This would be the case of geometrical optics or ray tracing, but rather the diffraction of light prevents it from getting smaller than a certain size, which is called omega naught, the spot size. That's the smallest spot size you can make for a laser beam for a given lens. And this is in many books. There's formulas for this. You don't need to worry. But what I'm going to tell you is there's something which is called the GUI phase. And this GUI phase, you can understand as the apparent phase shift between what would happen if there was no diffraction, in other words, geometrical optics, and the reality of physics, which is that there is diffraction, the Gaussian beam. And imagine that I have ray tracing, which means light at some point B will go in a straight line through the origin and come out here at this point E. But in fact, because of the diffraction of light, it doesn't do that. It cannot go through a mathematical point. Instead, it comes to this smallest distance here, C, and then diffraction forces it to expand again. It comes to the point D. And the point is that this path BOF is longer than this path BCD if you just measure the path length. And so a difference in path length means a difference in phase shift. So this apparent phase shift compared to the expectations of geometrical optics is called the GUI phase, invented in France, by the way. Okay, so we wrote some papers on this, and it turns out that there are spatial spectral couplings due to this GUI phase. And what this means is that the spatial distortions of the image and the spectral distortions I showed you before are actually intrinsically coupled. They cannot be separated. And what this means is that the image formation process in nonlinear optics is a lot more complicated than it is in linear optics. You cannot just see something in nonlinear optics and say, oh, there it is, that's its shape and that's its spectrum. It's more complicated than this. And this is a difficulty. So let me explain just briefly why this is the case. So what are we doing? We're focusing a laser to a small point, a very small focus, let's say a cubic micron with an objective lens. We're collecting a signal from that point, and then we're scanning the signal in space in 3 dimensions. And from this scanning, we reconstruct a 3-dimensional image by collecting the signal at each pixel, each voxel. So I want to differentiate between two types of nonlinear optical effects. There are ones which are called non-parametric and two-photon fluorescence, and something I'll introduce in a minute, SRS, which stands for stimulated Raman scattering. This is an incoherent process. So the signal generated in, this is my focal volume, right? This is my Gaussian focus. So there's the focal plane, and that's the divergence before and after the focus. So molecules at different locations, they are just emitting light randomly in phase compared to the other molecules. So the signal I collect by this big arrow here is the incoherent sum over all of these individual microscopic responses. Because it's incoherent, there's no interference between them. The total signal is just the sum of the individual signals, and that's it. I have to differentiate from what are called parametric processes, which are coherent processes, and that would be second harmonic generation, for example, or the Cars technique. In this case, essentially, each molecule is emitting a little bit of a laser beam in the forward direction. This, you remember this blue color we call the anti-Stokes? It's coherently emitted in the forward direction. And so the signal now is actually the coherent sum over this laser focal volume. And you can see, because it's coherent, a molecule on this side of the focus and a molecule on that side of the focus, they emit the same color and phase. They will interfere with each other. And as you know, interference can be both positive and negative. It can be constructive or deconstructive. So this is the origin of these artifacts, which I showed you in coherent non-optical microscopy, that you can get shadows, you can get spectral distortions. In non-optical microscopy, just because you see intensity in the signal doesn't mean that there's more molecules there. It's more complicated than that. There are coherent non-optical effects. So I'm going to introduce a different approach which gets around some of these problems. And this is called stimulated Raman scattering. It's essentially the first step of the Cars process, which I introduced to you yesterday. So the first step is I have a pump which goes up. There's no resonance there. That's a dashed line indicating that it's non-resonant. But I have a Stokes pulse which goes down, and the difference frequency between them is tuned to match some Raman mode. For the Cars process, there would be another interaction up and then a fourth photon down. But I'm just considering now the first step only. What you can see is that in this two photon process, this up and down process, that up means, if you like, a photon is annihilated or absorbed, and down means a photon is created or emitted. So every time this process occurs, I lose a pump photon and I gain a Stokes photon. This gives me idea of what's called a modulation transfer technique. If I modulate one of these two laser beams, it will affect the other laser beam because they're coupled by this coherent Raman process. So let me give you a more concrete example. Imagine that I have my two laser beams here. I'm showing them in orange and yellow, the pump and Stokes. And what I'm going to do is I'm going to modulate the red one, the Stokes beam. What do I mean by modulate? Literally going to put my hand in the beam, blocking it on and off. I won't really use my hand. I'll use an electro-optic device that can do this very quickly. But essentially, the pump beam is on all the time. This is the pulses, continuous stream of pulses from my pump laser. And the Stokes laser, what I do is every now and then I block it and I unblock it. So what you can see is that if I have the two lasers at the same time, this process will occur and I will lose a little bit of the pump light because it will be stimulated by this process. So you can see that when these are overlapped in time, the signal of the orange beam goes down a little bit. But when I block this red beam, the Stokes beam, this process will not occur because the red beam is not there. And so the orange beam, the pump, is at its normal level. And so by blinking this on and off, you can see I can modulate the output of the Stokes beam. So what I do is I measure the amount of power in this pump beam as a function of the Stokes beam being on or off. And I do this at megahertz frequencies. And so I use what's called a lock-in amplifier as a detection. I'm looking at the blinking frequency. And this signal here, this small signal, delta I, is proportional to this process. And this is called stimulated Raman scattering. It has quite a number of advantages. One advantage is that the signal is linear in concentration instead of quadratic. So when you go to lower concentration, that's good. It does not have this Fourier mixing background that the CARS process has because we're not measuring this blue color. We're just measuring the loss or gain in the transmitted beam. And we get, because it doesn't have this Fourier mixing background, we have a proper, correct Raman line shape. So I'll show you the comparison between the CARS and the SRS technique. So what I did is I took a molecule called DMSO and I diluted it 10 times in water, and I record the Raman spectrum using the CARS technique. And what you can see here is that I do have a Raman peak, but I've got this broad kind of background, which I mentioned also in my last lecture. So this is not great. This reduces our contrast. There's signal here, but it's not Raman signal. And that's a problem because it will look bright in your image, but it will not be from the molecule you're interested in. It could be from anything non-resonant. So this reduces contrast. Using exactly the same lasers, I don't change the lasers, the same lasers, same microscope, but instead of measuring the CAR signal, I use this modulation technique and measure the SRS signal, you get this. So everything's the same, but you can see now that the SRS technique has removed this Fourier mixing background. So now I really can assign the Raman spectrum, and it looks much more like the spontaneous Raman spectrum, but the signal is, because it's stimulated rather than spontaneous, the signal is at least 10,000 times bigger. So it retains that advantage. Let's go to still lower concentration. Instead of 10%, when we go to 1%, I will dilute another 10 times. So now the signal becomes very small. And you can see that in the CARS technique, there still is signal, but it's non-resonant. Maybe somewhere under here, there's, if you have some imagination, maybe there's a bit of a Raman signal, but we have to admit that it's hard to see. What about the SRS technique? Well, the SRS technique still can see the Raman peaks, but there seems to be some kind of background there as well. So it's not perfect, it's much better, but there still remains some background. Let's go to lower concentration still. Let's reduce the concentration by another factor of 2. And this is using the SRS technique. So you can see that the Raman peaks are there, but there's some kind of background which is growing in as I drop the concentration. Let me remove my molecule completely and just have pure water, which should be non-resonant. And you can see that there still is a signal there. It's non-resonant. So what can that be? Why is there a signal even though there's something non-resonant? This is a problem for the following reason. This is from a paper we wrote a few years ago where we're looking in geophotonics. We're imaging a mineral called calcite inside other rocks. And calcite has a well-known Raman spectrum. It has a resonance around 1100 wave numbers. And it's seen here in this red peak. But you can see in the image, this is a white field illumination. This is some piece of rock that has some minerals. And you can see when I do my SRS imaging technique, you can see that it looks bright in various places. And you would think, oh, well, when it looks bright, that must be more calcite. And where it's darker, there'll be less calcite. But that's not correct. And the reason why it's not correct is because there's other types of background signals, and some of these can be quite large. The reason I know it's not correct is because in each of these pixels, I can scan the Raman spectrum. In this region here, I scan the Raman spectrum and I get a peak, which I know is calcite. But when I scan the Raman spectrum here, I get this big blob. So I know that's not calcite, maybe a little bit, but not mostly calcite. So there's an issue here. Just because the image intensity is bright doesn't mean that that's the molecule that you're imaging. There are other effects that can make the image look bright. And so that's something to worry about. You would misinterpret this image if you thought that wherever it's bright, there's more calcite. So why are there signals? Well, let's think about the basic physics of what we're doing. We're sending two laser beams focused into a sample, the pump and the Stokes. And one of them I'm blinking on and off, I'm modulating. And what I'm doing is I'm looking for how the presence of one laser beam affects the transmission of the other. If there's any coupling which affects it, then there will be the modulation will be felt by the other laser beam. And this is what we measure in SRS, is this transfer of modulation from one laser beam to the other. So this means that any type of physical coupling that couples them into the focal volume, in other words, anything where the presence of one laser beam affects the other laser beam will appear like a signal to you. So what could these be? Well, here's one example of nonlinear absorption, where you could have a non-resonant or resonant term where the presence of one laser beam, the green one, allows the yellow one to be absorbed. But if the green one wasn't there, then there would be no absorption. So if this happens, this would be a loss for both pump and Stokes, but you can definitely see that when one laser beam is present, it affects the transmission of the other. This is a nonlinear absorption type of coupling. But there's another type of coupling, which is a bit more subtle, and it's called a nonlinear scattering term. And essentially, it's where the presence of one laser beam affects the divergence of the other laser beam. In other words, the presence of one laser beam looks like a transient lens. There's two types of transient lens. One of them is called cross-phase modulation, and the other is called thermal lensing. These things are well known in optics, and what it means is that your detector, which is somewhere in the forward direction, this is collecting the light from the focal volume, which goes forward to your detector. It's almost always the case that we cannot collect all of the forward-going light. We collect some solid angle. So if there's a lens effect by one laser beam that affects the propagation of the other, if the lens is negative, meaning that the uh, light is more diverging, then less light will fall on your detector when the induced lens is there, and this will look like a loss. On the other hand, if the lens was positive, it will be like a little bit more focusing and more light will fall on the detector, and this will look like a gain. So these transient lens effects, these nonlinear scattering terms, can also appear as a modulated signal, but these are not Raman processes. These are non-resonant. So these terms are background signals also in SRS spectroscopy. They're the origin of this bump that I showed you, which reduces our contrast and reduces our selectivity. So we would like to understand these, and if we can understand them, we would like to get rid of them so we can have a more sensitive and more specific microscopy. What I can tell you is that all existing modulation techniques have some background because there's these various physical processes which really exist. I'm going to show you, though, that we came up with one which, in fact, I hope to convince you, removes all these backgrounds. And I'll show you this idea. I told you that for the case of DMSO in water, we seem to have this background, and we see it even in pure water. Pure water doesn't absorb, not at the wavelengths we're using. 900 nanometers for the pump and 1030 nanometers for the Stokes. Water is transparent. So water is not absorbing. There's no two-photon absorption. There's no excited state absorption. Because there's no absorption, there can be no thermal effect because that requires light to be absorbed. So the only term left is what's called cross-phase modulation. So we believe that this term that is coupling the two beams together in pure liquid water is cross-phase modulation. And this is relevant to biological imaging because, as I think we agreed last time, biology is full of water. So these things will happen whenever there's water. Okay, without going through the physics, what we're going to do is a new kind of polarization modulation. So instead of an amplitude modulation where I have one laser beam on and the other one I'm blinking on and off, what I'm going to do is I'm going to flip the polarization of that laser beam, parallel, perpendicular, parallel, perpendicular. And I'm going to modulate in this way. And you can see that there's an interesting aspect of this, which is I'm not changing the total laser power through the sample. All I'm doing is flipping the polarization. But there are selection rules in Raman spectroscopy which depend on polarization. And so what I'm doing is I'm modulating between parallel and perpendicular transitions. So this works, and I'll show you the idea. I showed you this before. This is comparing the cars with the amplitude modulation, SRS, stimulated Raman scattering. Amplitude modulation where I'm simply turning the other laser on and off. And that's the non-resonant background in cars. If I lower the concentration and I do amplitude modulation SRS, I get the Raman peaks, but I have this background, which I argue to you is due to cross-phase modulation. If I do the polarization modulation, this is removed. So you can see that now, even though it's at low concentration, I really have pretty close to a flat line. So I have this cross-phase modulation background, which is present in this SRS image, but with polarization modulation, I can remove it. So this looks quite promising, but there are some limitations to the polarization modulation technique. It removes one type of background, the cross-phase modulation. It has all the advantages of spectral focusing and scanning and so on, but it only works for certain types of Raman bands that are so-called parallel. And so it works in some cases, but will be limited. There are other cases where it will not work. So we would like to find a more general solution than this polarization modulation. So my question is, can we remove in some simple way all of these non-Raman backgrounds? Can we remove the cross-phase modulation, the thermal lensing, and the nonlinear absorption? But we don't want to pay a big price. We would like to keep the very nice, easy Raman spectroscopy that we developed, this spectral focusing, this tuning just by changing the time delay. These are very nice features. And so we would like to keep that because that's a big advantage. So in other words, we need to develop a new type of modulation scheme that removes these backgrounds, all the non-Raman terms, but keeps the Raman term. Can we come up with an idea that will allow us to do that? A new kind of modulation, not amplitude modulation, not polarization modulation. So I'm going to give you a clue. And a clue is, if you remember in the Raman spectrum, that the Raman resonant terms are sharp peaks. In other words, the Raman response varies rapidly with frequency. They have sharp features. But the non-Raman terms you saw very slowly. They look like a broad bump. They don't have sharp features. So there's a clue in whether something responds quickly or slowly to the Raman resonance frequency. Let me show you this with an illustration. So this is going to be a little calculation. Yesterday I showed you how we record the Raman spectrum, and that is we have a pump and a Stokes beam. We stretch them in time. We chirp them. And when we chirp them, the instantaneous difference frequency becomes narrow. That's this projection onto the frequency axis becomes very narrow. And as I change the time delay, I can tune this frequency difference. And that's how we do this, what's called spectral focusing Raman spectroscopy in the microscope. So here's a calculation. I have two resonances, one very narrow, one broad. And there's something resonant in my sample that would give a response like that. Okay, so those are the Raman resonant terms. What about the non-Raman terms, like cross-phase modulation, thermal lensing, etc.? What would they look like? Well, I would do the same thing. I would scan them in time delay, just like before, but there's no Raman resonance. So what the signal would look like would be essentially a cross correlation between the two laser beams. It go up and down as the two laser beams pass through each other when I scan the time delay. So that looks like this background that I showed you. Of course, a given sample can contain both the Raman and the non-Raman terms. So if I do them both at the same time, I could get something that looks like this. And if you remember, this looks quite a bit like the data I showed you, where I have some sharp peaks that are embedded in a broad background. So that's the problem we have. How can we separate the sharp peaks from the broad background, which we don't want? The broad background is not specific to the molecule we want to study, that we want to image, and it reduces our contrast. Okay, so can we find a modulation scheme which separates the terms that vary rapidly with frequency from the terms that vary slowly with frequency? But I have a restriction, quite a severe one. Essentially, don't want to change anything else about my laser pulses. I don't want to change the amplitude, the intensity of my laser pulses, because if I do that, then I would be doing amplitude modulation. I don't want to change the pulse duration, because this would change the intensity of the pulse. I don't want to change the polarization. I don't want to change the spatial mode, because all these things would amount to some other kind of modulation, and I would introduce some kind of background. So can I have some modulation scheme which separates the fast from the slowly varying terms, but does not change anything else about the laser pulses? It seems hard to imagine. Well, there is a way, which I will tell you about. But first, I'm going to make an analogy. Um, some of you may use Photoshop or some image processing techniques. In Photoshop and other methods, there's something which is called the unsharp mask. And it's basically, I'm going to make an analogy with a spatial frequency filter. So the idea of an unsharp mask is I have an image, an original image, and what I'm going to do is I'm going to blur it with a Gaussian blur. I'm going to blur it out, and then I'm going to take the original and I'm going to subtract off the Gaussian blurred. And you can see what I end up with is just the sharp edges. So this is a technique for sharpening edges in imaging. If you play with Photoshop, you'll see that. So the unsharp mask is the original convoluted with this Gaussian blur, which is the original, the sharp minus the blurred, the unsharp, and we get that. So can we use this idea to make an analogy in the Raman spectral domain? Because here we have something which is sharp, meaning it varies rapidly with space, and here's something smooth, which means it varies slowly with space. Can we use this idea in the Raman spectral domain? And it turns out that we can, and I'll explain as follows. This technique is called chirp modulation, SRS, and you can think of it like an unsharp mask in the frequency domain. So you remember I described to you how we do our Raman spectroscopy. We have the pump and Stokes, and we give them the same sign of chirp, both a positive slope here, such that their difference frequency is narrow. Difference frequency means the distance between these two pulses is constant, and this gives a narrow band frequency. Well, what if I did this? What if I just flipped the sign of the chirp? Instead of a positive chirp, I gave a negative chirp. So everything else in my laser pulse is the same. The pulse duration is the same. The intensity, everything's the same. All I did was change the sign of the chirp. In other words, when I have a positive chirp, this one, it means that the red colors come first and the blue colors come later. Remember, chirp means that a variation of the instantaneous frequency during the pulse, like a Doppler effect. All I'm doing here is I'm reversing the sign. Here, instead of the red coming first, I'm making the blue coming first. But pulse duration, the spectrum, everything remains the same. So what are the consequences of reversing the sign of the chirp? Well, now you can see that the difference frequency is no longer narrow. The difference frequency, the instantaneous distance between them, is now broadband. So this instantaneous difference is now very broadband. So if I modulate between the co-chirped and the contra-chirped, in other words, I modulate the sign of the chirp, then essentially what I'm doing is I'm modulating the Raman spectral resolution between high resolution and low resolution. I keep the intensity constant, the spectrum, the polarization, the spatial one, everything else is the same. I'm just modulating the sign of this chirp. And it turns out that this technique essentially removes all of the non-Raman background terms. And I'll show you how this works. Okay, so here's the analogy with my sharp mask. I have my original image, which is a sharp mask. And I'm going to say that this is analogous to this co-chirped technique, which was what we use for amplitude modulation. So this is actually real experimental data. So this is 10% DMSO in water, and I get these two Raman peaks that I showed you before. I'm going to do the contra-chirped, which is like the Gaussian blur. It's a low resolution. So when I do that, I lose the spectral, I cannot resolve these things, and everything is kind of broadened out. The idea of the unsharp mask is I take the original and I subtract off the blur. So what I'm going to do here is I'm going to take the co-chirped Raman spectrum, I'm going to subtract off the contra-chirped, and what I get is this. It has a bit of a funny shape, but you can see that the Raman peak is still there. Why is this interesting? Well, what I'm going to do is I'm going to keep dropping the concentration lower and lower. So on this column here, I have the co-chirped. As I drop the concentration from 10% to 1% to 0.1%, this is a factor of 10, another factor of 10. And you can see in the AM amplitude modulation co-chirped, the signal goes down, 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 and I'm dominated by the non-resonant background terms. In the contra-chirped, it does the same. But the chirp modulation is hopping back and forth between these two and subtracting them. That's the modulation. And you can see that even though the signal looks like it's not there, you can see that even at the lowest concentration, there still is a Raman peak. So this shows that I can go to much lower concentration than I ever have before, and I can still identify the Raman peak. So this is important. In fact, what I can tell you is this is now the Guinness Book of Records for sensitivity in SRS microscopy. And I'm showing, and this is a logarithmic plot showing the signal as a function of concentration in a log-log plot. So you can see that the amplitude modulation and the chirp modulation look both linear at high concentrations, but as I go lower and lower and lower, you can see that the amplitude modulation, the blue dots, they deviate. Why? Because they start to contain more and more contributions from these non-Raman background. But the chirp modulation does not have this non-Raman background, and so I can follow it all the way down to the lowest concentration. And this has now been patented, and we hope that this will be becoming commercially available to microscopists. So the line has a funny shape. Can we deconvolute this funny line shape to get a proper Raman spectrum? Turns out we can, because this is a linear convolution. This unsharp mask idea is a convolution of the amplitude modulated signal with this blur, if you like, the sharp minus the blurred. And this is the so-called chirp modulation spectrum. That's what we measure, and this is how it comes about. It's this convolution. If I know the convolution, then I can do it in the reverse way. I can do a deconvolution to recover the unperturbed Raman spectrum. So this is the Raman spectrum of the co-chirped. Here it is, the blue. The orange, which I'm going to flip in sign because I'm going to subtract it, so it's going negative, is the contra-chirped. And when I subtract the two, I get this chirp modulation. You can see, because of the subtraction, you can see that the gray signal, which is the chirp modulation signal, it's got a little bit of funny line shape, which dips below zero, comes back up, and goes below zero again. But the reason why is because of the subtraction. But everything is linear, so I can work out what's called the convolution kernel, which I need for the reverse transformation, the deconvolution technique. So this I can measure by independently measuring the co-chirped and the contra-chirped. I get a kernel for each, and the chirp modulation is just the difference between these two. So I can get this kernel, and with that kernel, I can then do a deconvolution. So without going into details, I can show that this works. And here's an example where I have in blue the spontaneous Raman spectrum, and in orange I have the deconvoluted chirp modulation Raman spectrum. And actually I had to displace them from each other so you can see the difference because they're almost identical. So what this means is that the deconvoluted chirp modulation Raman spectrum really gives almost identically the spontaneous Raman spectrum, but now it's tens of thousands of times more sensitive. So this looks quite promising. Can we test this idea? So we're going to do something that's really difficult for coherent Raman techniques, is we're going to look at plant material. Why are plants difficult to image in biology? It's because they're often very colorful. Colors mean absorption. Absorption means that there will be lots of other nonlinear terms that will interfere. So this is an example of an image of a piece of plant material which is highly absorbing, highly fluorescent, and it's also highly scattering. So this would be a big problem for ordinary uh, microscopy. So what I'm going to show you is I'm going to show you a series of images where I compare amplitude modulation, the industry sort of standard technique for stimulated Raman scattering microscopy. And what I'm going to do is I'm going to change the Raman shift, and I want to see how the image changes, because I would expect when I change the Raman shift, I would be probing different molecules within my sample. So amplitude modulation at this Raman shift and these two other Raman shifts, and I don't know if you can see very well, but these images essentially look the same. That should worry you, because what it means is that these are dominated by non-Raman terms. They don't care what the Raman shift is. They give some signal independent of the Raman shift. Whereas with the chirp modulation, you can see that as I vary the Raman shift, that the image does indeed look different. This is a good news, because what it means is it's sensitive to the chemical composition of the sample. There are different chemicals in these different images. So I can put these together with a false coloring. And if I do it for the amplitude modulation, essentially I get a grayscale. But with the chirp modulation, I can color these with an RGB color scheme. And you can see essentially now as I look in color, I can really identify these different portions inside the sample by their Raman spectrum. So this really shows how it works. In fact, to prove it, I can show you that there are different Raman spectra for these different regions here. So it really does work. Let's do something even more challenging. We're going to take a sample from what in English is called a sweet potato, this orange colored potato. And it contains compounds that are called carotenoids. And these are very, very strongly absorbing. And this has been shown to be almost impossible to study by Raman spectroscopy and microscopy. When I do the amplitude modulation technique, I get brightness everywhere. When I do the chirp modulation, I see that the image looks different. So in fact, the chirp modulation techniques allows me to identify these different carotenoids. This has never been done before, actually. A sweet potato may not be the most important problem in the world, but what it shows is that even in very strongly absorbing samples, that we can get the Raman information, which allows us to identify that there's two different types of carotenoids in the sample. Let me show you a more important example. So I have a colleague at the University of Ottawa that was interested in this field called cryopreservation. You may know that there are some frogs and some fish that hibernate in the wintertime, certainly in Canada, and they are frozen solid every winter. And when the spring comes, they thaw out and they hop away or they swim away, and they're perfectly fine. You cannot do this with a mouse. If you put a mouse in the freezer and you take it out in the springtime, that mouse will not run away. That mouse will be dead. And the reason why is because the ice crystals that form when something is frozen essentially break all the cell membranes and basically break the cells apart. This is why frozen meat, frozen fish does not taste the same as fresh. It's because the ice crystals have broken all the cell membranes. These frogs and these fish have a special antifreeze protein that prevents the ice crystals from forming. In other words, the water undergoes a liquid to glass transition rather than a liquid to crystal transition. And this means there's no change in volume. As you know, ice is larger than water. This is why ice cubes float. So If you could somehow take advantage of these antifreeze proteins, then you could freeze human tissue as well. This would be of huge importance in biology and medicine because if someone needs, God forbid, a kidney or liver transplant, then they have to find someone within 3 days or so who can offer one for transplant. And the reason is because you cannot store a liver or kidney in the freezer because it would no longer be alive. So cryopreservation was a very important idea. So this colleague is using molecules that look like this, and this is similar to the molecules that the frogs and the fish use to keep ice from forming. But there's lots of questions that they don't know about, like how does this cryopreservation work? Does this molecule go inside the cell, or does it go outside the cell where there's water as well? So they wanted to know, and so we said, well, we can do our SRS imaging because it's so sensitive. And what we did here is we looked at a single cell, and we want to apply this drug and to see if we could see how the drug penetrates a single cell. So this would be called single cell pharmacokinetics. You could imagine this would be the ultimate approach to drug discovery because you could make a movie of how a drug penetrates a single cell. So I wanted to show you that actually this works. This is the first demonstration of single cell pharmacokinetics. And what this showed is that we can track the concentration inside the cell as a function of time. And what this shows is that actually their cryopreservation drug does indeed go inside the cell. It takes some hours, but it does go in. And this is a plot of the concentration of the drug inside the cell as a function of time. So that's a demonstration of what can be done when you have this enhanced sensitivity and contrast from this chirp modulation. So with this, I will conclude. Um, I showed you that there are background signals in stimulated Raman scattering. The new modulation technique I introduced, which I call chirp modulation, removes these backgrounds, but it retains the facility and the rapidity of the spectral focusing technique. It removes all of these non-Raman backgrounds, and I believe it is the world record sensitivity in SRS microscopy. I also showed you that using a deconvolution, you can obtain the spontaneous Raman spectrum, and importantly, the signal linear in concentration. And I showed you a first example in single cell pharmacokinetics. And we hope that this technique will catch on and people will apply it to all kinds of problems, not only in biology and medicine, but hopefully in other fields as well. So with that, I'll end and I'll be happy to answer any questions.